Hello everyone, I'm back with another video. This is Dr. Visha and today we are going to be solving our paper 2 O-Levels Biology, October-November 2023, 5090. And as you all know that this paper consists of 7 to 8 questions and it has 8 marks, and the time duration for this paper is 1 hour and 45 minutes. Alright, so let's begin solving this paper. So the question number 1 says that cholera is a type of organism that can be passed from one host to another. State the term used to describe a disease-causing pathogen. We all know that anything that causes, a disease-causing organism. I just said the answer myself. But anything that causes a disease or that initiates an immune response in a human body is known as pathogen. Alright, the type of the disease in which the disease-causing organism is passed from one host to another. We all know that the disease in which the disease or causing organism is passed from one host to another host is known as a communicable disease or an infectious disease. So, without wasting any more time, we move towards part B, which says that a disease-causing organism that causes cholera is a bacterium. What is it? It is a bacterium. Alright, so let's highlight this keyword that the disease-causing organism is a bacterium. Complete the 1.1 to list some of the structures present in a bacterial cell and the function of each structure. So they are assessing your knowledge on the structure of protozoa or the structure of bacteria. So the ribosomes, function of the ribosome, we all know that they are for the protein synthesis. Then what is the structure which has a function of having small rings of DNA that carries genes? They're the small rings of DNA that carries genes. They are actually called as plasmids. Alright, then what controls the passage of substances into and out of the cell? So we call it cell membrane or a membrane. Alright, then there is the cell wall. What is the function of cell wall? Obviously, the function of the cell wall is to provide the structural support or to provide the shape to the cell. Alright, so this gave you 4 marks. Then part C says figure 1.1 is a photograph of a bacterium that causes cholera. So this is a bacterium which causes cholera. It has a body, it has a plasmid, and it has a tail-like structure which is known as flagellum. So they say that the length of bacterium in a photograph is 100 micrometers. Use this value to calculate the actual length of the bacterium in micrometers. So the length of the bacterium in the photograph is 100 millimeters. Let me highlight this. The length of the bacteria is 100 millimeters, and we have to use this value to calculate the actual length of the bacterium in micrometers. So first of all, we need to convert this 100 millimeters into micrometers. So 1 millimeter equals to 1000 micrometers. So obviously 100 multiplied by 1000 will make up to 100,000 micrometers. Alright, so what will be so they are actually asking the actual length of the bacterium. So actual length equals to the image length over the magnification. So the image length was 100,000 micrometers divided by what was the magnification? Magnification is given over here, that is 50,000 micrometers. So by doing on the working, you will get 2 micrometers as the actual length of the flagellum of this bacteria. Alright, so I hope this was clear. Let's move on to the next part of this question, that is, use your knowledge of a type of specialized human cell to suggest the name of the structure Z and to state its function. So the name of the structure Z is, we already discussed this, that is the flagellum. And what is the purpose of the flagellum? It provides motility or the movement or the locomotion to the organism. So the function is locomotion. Alright, so part 3 says the binomial name of this type of bacterium is Vibrio-cholera. State the genus of this type of bacterium and one advantage of the binomial system of the species. So the genome is going to be always remember that the first part of the name of a species is known as its genome. So what will the genome is underneath of Vibrio, and the advantage for using the binomial system that it is an international international or standard system provides these to work and study species around the world. Alright, for part 4, explain how the presence of bacteria of this type in a human causes diarrhea, one of the symptoms of cholera. So they're asking that how do these bacteria causes diarrhea, which is a symptom for the cholera. So every bacteria has a toxin, we all know that, right? So the bacteria is going to release a toxin, and what will they do? The toxin will cause increased permeability of the intestine to the chloride ions. All right. So what will happen? The chloride ions will increase in the intestine, and obviously, what will they do? They will lower the water potential of the intestine. So the water will not be absorbed from the intestine, and the water is going to move into the intestine, and this will going to increase the motility of the intestine. So basically what is happening over here, osmosis is happening over here. The water is coming inside the intestine through osmosis and increasing the motility of the intestine, causing the diarrhea. So I hope that was clear. So this is the answer that I have written for you, so you can read it out from here. So we have to give 4 distinguishable points, and we have to tell the etiology or the pathophysiology of the diarrhea in the cholera. So this was the answer to this question. Bacteria produces a toxin, which results in the chloride ions to increase in the intestine. That lowers the water potential of the intestine, and osmosis takes place, and water comes inside the intestine and causes the frequent stools. All right. So now, question number 2. Souls are reindeer. The reindeer live in areas of Arctic where there are no trees. In some months of the year, the reindeer eat mostly grasses, ferns, and mosses. Reindeer are eaten by wolves and brown bears. Construct a food web to show the flow of energy between the organisms in this ecosystem. So I have drawn the food web for you. So this is a food web. Basically, what happens is that the producers, what are the producers? Producers are the organisms which can make their own food. For example, in this, grasses, ferns, and mosses. Grasses, ferns, and mosses. Alright, so this is the first trophic level. What is the first trophic level? Which are the producers? So these are the producers. So we added them in the first trophic level. Then the energy is transferred from the grass, fern, and mosses, means from the producers to the consumer, that is a reindeer over here. Alright. So this is the reindeer. All of this arrow shows the flow of energy from the producers to the consumer, that is reindeer, and then the reindeers are eaten by the wolves. The energy present in the reindeer is getting transferred to the wolves and the bears. Alright, so this is why we have the arrow pointing from the reindeer to the wolves. So this is the second trophic level, and then the third trophic level is the final consumer of the food. Alright, so you don't need to write down or label the trophic levels. I just added these labelings to explain you and to revise the concept of trophic levels and food webs. You just had to draw the food web like this and make sure to use the 2/3 of the space provided in the question. Alright. So part B says, in other months of the year, reindeer eat an alternative food source. This alternative source of food contains a carbohydrate called lecithin. Reindeer are the only animals that are able to digest lecithin. List the chemicals element that make up a molecule of lecithin. So, what are lecithin? Lecithin is a carbohydrate as mentioned in the question. Alright, lecithin is a carbohydrate. So, what are the components of the carbohydrates? We all know that carbohydrates are made up of carbon, hydrogen, and oxygen. So, carbon, hydrogen, and oxygen. Alright. So, part 2 says there is one advantage to reindeer of eating food that contains lecithin. What is the advantage of to the reindeer by eating the food that has lecithin? So obviously this is going to provide energy, and it can be converted to the glycogen. Alright. So you must have noted one thing in my answer, that I have written the word provide. I have not written the word produce or like release, stuff like that. So you need to make sure and you need to take care of the terminologies you are using in the answer. That should that should resonate with the mark scheme, or that should resonate with the demand of the examiner has from the students. All right. So let's come towards part three. That is, your knowledge of how cell function is controlled suggests why other animals are not able to digest glutenin. Obviously, when the animals glutenin is a carbohydrate, so obviously we need enzymes to digest glutenin. So why other animals are not able to digest glutenin? Because they must not be having the enzymes that are required to digest the glutenin. And it carries two marks. So we need to provide 2 points. Number one is that there are no enzymes. Number second is, why do they don't have any enzyme for the glutenin? Because they don't have the genes that are present to that should be present to code for the enzyme for glutenin. So this is the answer to this question. That they don't have the enzyme to digest glutenin, and they don't have the genes that code for the enzymes. For the enzyme to digest glutenin. Now, for question number three. Figure 3.1 shows diagram that represent the blood flow into and out of the three major organs through blood vessels P to U. So this is the figure. I have labeled it for you that the blood vessel that is coming towards the lungs is pulmonary artery, and that is leaving the lungs is pulmonary vein. The blood vessel that is coming inside the kidney is renal artery, and the blood vessel that is leaving the artery is renal vein. The blood vessel that enters the liver is hepatic artery, and the blood vessel that leaves the liver is hepatic vein. So we need to identify each of the following blood vessels. Q is the pulmonary vein. R is the renal artery. You need to write a full form while attempting an exam paper. Then U is hepatic vein. Hepatic vein. It's possibly self-explanatory. Now, part B says compare the structures of blood vessels Q and R. So what are Q and R? Q is the pulmonary vein and R is the artery. So we need to compare the structures of artery and a vein. So we all know that the arteries have thicker walls, thick and muscular walls, and they have more elastic in their walls, right? Moreover, they have narrow lumen and they have no valves. So these are the structural properties of the artery. And if we talk about the structural properties of the vein, so the vein is comparatively thinner, the vein has comparatively thinner walls than the artery, and they have thin layer of muscle and elastic tissue in their walls. Moreover, they have the wide lumen and they have the valves to prevent what? To prevent the backflow of the blood. All right. So the answer to this question, while writing down the comparison and the differences or the comparison, we need to make sure that we are talking about the two particular things we are comparing and what we are writing the differences between side by side. Like for example, if I talk about walls of both veins and arteries, I'll talk about both of them at the same time. All right, I'm not going to do that like. I'm not going to write everything about veins first and then write everything about arteries first. No, this is not the way to answer the sort of question. Alright, so let's move on to part C. Explain each of the following. Blood vessel S has a lower concentration of urea than blood vessel R. So what is S and R? S is renal vein and R is renal artery. So they are saying that S has lower concentration of urea than blood vessel R. So why is that so? This is because urea is entering with the blood into the kidney through the renal artery, and then kidney removes the urea through filtration, and then the urea is excreted in the urine. So the blood that's entering entering the kidney through renal artery has more urea as compared to the blood leaving the kidney through renal vein. Why is that so? Because the kidneys are filtering the urea, and the urea is excreted in the urine. So what are we going to write down? That urea is entering in the kidneys through renal artery. Kidney filters the blood and urea is excreted in the urine. So this gives us 3 marks by telling urea is entering in the kidneys through renal artery, 1 mark for this. Second mark for this, that kidney filters the blood, and third mark for telling the urea is excreted in the urine. Part 2 says blood in vessel U sometimes has a higher concentration of glucose than blood in vessel T. So what is U? U is hepatic vein and T is hepatic artery. So they are saying that blood in blood vessel U means in hepatic vein. Hepatic vein has higher concentration of glucose than hepatic artery. So why is that so? Hepatic vein is having higher concentration of glucose than hepatic artery because blood glucose, like body needs more sugar, right? So to compensate the need of the body for the sugars or for the glucose, glycogen is getting broken down to glucose, and then glucose is passed out of the liver through U, that is hepatic vein. All of this process is stimulated by the glucagon. So the answer to this question will be that body needs glucose to glycogen is broken down to glucose. The glucose is passed down out of the liver into the hepatic vein, and this is stimulated by glucagon. So this was 3 marks, and we gave points to this question. Now, question 4 says, figure 4.1 shows a Siamese fighting fish that lives in fresh water. It has large, colorful fin. Wait. So it has large, colorful fin and location of an organ that can be filled with atmospheric air. So they're talking about gills. So this area is the gills. All right. So figure 4.2 shows the effect of what are they showing? They're showing the effect of changing pH on the solubility of oxygen in water. So the change in pH on the solubility of oxygen in water, effect of changing pH, right? So the next axis they have plotted pH and the percentage solubility of water and oxygen. So we can see that when the pH increases, the percentage solubility of oxygen in water is increasing as well. So figure 4.3 shows the effect of changing temperature on solubility of oxygen in water. So one graph is telling the effect of changing in pH. Another graph is telling the effect of changing in temperature. As the temperature changes, the solubility of oxygen in water is decreasing. Okay, so Siamese fighting fish live in shallow water that can range in pH from 6.9 to 8.2 and at temperature from 15 to 40. So they can live in the pH from 6.5 to 8.2 and temperature ranges from 15 to 40 degrees. Use this information to explain, like in figure 4.1, figure 4.3, to explain how conditions in which of these fish may be challenging. So the best thing and the best way to approach this question is to look at the graphs again. So basically what the question is asking us, question is saying that explain how the conditions in which these fish live may be challenging. So if we talk about figure 4.2, we can see that at figure 6.9 oxygen availability is low and at 8.2 oxygen availability is high, and this is a range in which the fish can survive. Alright, so below 6.9 oxygen availability is very low for the fish to live or to carry out aerobic respiration, right? In the same way, if we look on to the graph of temperature, so we can see the temperature range which was given in the question was that the fish are able to survive in the temperature between 15 degrees Celsius to 40 degrees Celsius. All right, so 15 degrees Celsius till 40 degrees Celsius means this is the range at which the fish can survive, not more than this and not less than this. All right, so what are we going to write down in this question? That there will be low oxygen availability for the fish at pH 6.9. See, 6.9, low oxygen availability, and at the temperatures temperatures below 40, like temperatures above 40 degree. All right, so the low oxygen availability at pH 6.9 and 40 degree Celsius for aerobic respiration. All right, so the second thing we have to write that the pH, as the pH is increasing, the solubility of the oxygen is also increasing, and as the temperature increases, the solubility of oxygen is decreasing. All right, so this is the answer to this question that you have to write, that low oxygen availability for fish at pH 6.9 and 40 degrees Celsius for aerobic respiration. As temperature increases, the solubility of the oxygen decreases, and as pH increases, the solubility of oxygen increases. All right, so one thing I want to mention over here is that data code carries 1 mark. Either you can code the data from the pH graph, or you can code the data from the temperature graph. So if we talk about pH, you can see that at pH 6.5, percentage solubility of oxygen is around 63%, and at pH 8.5, the percentage saturation of the oxygen is around 98%, as you can see over here. All right. If we talk about temperature, then you can quote that at 10 degree Celsius, the oxygen availability is around 11 or 11.3. Yes, 11.3 um, mg per dm cube. And at 50 degrees Celsius, the oxygen availability is as less as 5.4 milligrams per dm cube. All right. So you can quote the data either from the graph of temperature or from the graph of pH. The important thing is that you use the correct units. All right. So data code carries 1 mark. You can make sure that you score full marks. Coming towards part 2 of this question, which says that all fish have gills that provide a gas exchange surface in the water. Some Siamese fighting fish also have an organ that has similar features to those of a human lung. These fish swim to the surface of the water to take air from the atmosphere into this organ. Explain the advantage of Siamese Siamese fighting fish of also having this organ. Basically, which organ are they talking about? They're basically talking about the gills, which are the organs like lungs in the bodies of the fish. So they are useful in the low oxygen conditions or the no oxygen conditions, and the gaseous exchange between the body of the fish and the air, and make the more oxygen available for the fish for aerobic respiration, which enables them to release energy or make use of the energy. All right. This is the answer to this question, that they are important and are useful in the low oxygen conditions. They make sure that there is adequate gaseous exchange between the body of the fish and the atmosphere, and more oxygen is available for the fish to carry out aerobic respiration to release energy. All right. Now, part B says Siamese fighting fish have 42 chromosomes in each diploid cell. State each of the following. The named structure in each diploid cell that contains chromosomes. We all know that nucleus contains every type of genetic material. All right. Then the type of nuclear division that produces gametes. We all know that meiosis produces gametes. Then the number of chromosomes in each gamete of the Siamese fighting fish. So Siamese fighting fish have 42 chromosomes in each diploid cell. This is a very important line you should notice, and you should highlight that they have 42 chromosomes in each diploid cell. Means diploid cell has 42 chromosomes. So the gamete are the haploid cells. So the gamete will be having single pair of chromosomes or single chromosome, right? So 42 divided by 2 makes 21. So the gametes will be having how many chromosomes? They will be having 21. All right. So I hope this was clear. Then part C says the dramatic shape and color of the Siamese fighting fish are produced by selective breeding. So just how the process of selective breeding in Siamese fighting fish can be used to produce these desirable features. So what is the desirable feature over here? The shape and the color of Siamese fighting fish. Alright, these are the desirable features or the characteristics. So they are produced by selective breeding. What is selective breeding? Selective breeding is basically the artificial selection that is done by the humans, in which they select the organisms or the population with their desirable characteristic, desirable features. So this means that the fish having dramatic shape and color will crossbred with each other for several generations. And how much time? Till the time where all of the fish would develop the desirable characteristics. All right. So you're going to write the answer like this: that you can select the fish with desirable features and they crossbreed the offspring with desirable features, or the best features are selected again to further breed, and this is repeated for many generations. All right. So let's move on to the question number 5, which is the next question. A student investigates the use of enzyme in fruit juice production. So let's highlight this. What is the investigation about? Investigation is about the use of enzyme in fruit juice production. The student carries out the following steps. Cuts one apple into small pieces, puts the same number of pieces into each of the 3 beakers A, B, and C. Add 2 centimeter cube of a dilute enzyme solution to the apple and beakers A and B, and add 2 centimeter cube of the distilled water to the apple in beaker C. So they added 2 centimeter cube of the juice in 2 beakers and 2 centimeter cube of the water in 1 beaker. Places each beaker in a water bath at a constant temperature of 30 minutes. Filter the juice from each beaker into separate measuring cylinder and record the volume of the juice collected in each measuring cylinder. So identify the enzyme that the student should add in the apple beakers A and B. So first of all, let's understand the investigation. What did they do? They cut the apple into small pieces and they added the apple into the beakers, 3 beakers, and in 2 beakers they added the dilute enzyme solution, and in 1 beaker they added the distilled water. So the enzyme would act on the apples and will produce the juice. So apples have the cell walls, and obviously the enzyme that will be required to digest the apples, the cell wall will be known as pectinase. All right. So the enzyme will act on the pectin and will convert the pectin and help the breakdown of the apples into the emulsified or juice form. All right. So what will be the enzyme? Pectinase. Outline the reason why students set up beaker C. So in beaker C, they added distilled water. They added nothing. So we always in every investigation, we have the control. We set up a control to make a valid comparison. In which we either add a dead organism, dead plant, dead animal, or the distilled water. So control is for the, this is a control to make the comparison. I hope that was clear. Now, part B says the results of the students' investigations are shown in table 5.1. This is table 5.1, beaker A, B, and C, temperature in degrees Celsius, volume of juice collected. The temperature was at 45, 20 cm cube was collected, at 90, 5 cm cube was collected, and 45, 5 cm cube was collected. At 45 it was 20, and as the temperature increased till 90, it got decreased till 5. And if after that, even if they are talking about the beaker C. The beaker C was kept at 45 degree. That is the optimum temperature it was running. All right. So use your knowledge of enzyme action to explain these results. So we can write down that beaker A produced more juice than beaker B at 45 degrees Celsius as compared to beaker B that was at 90 degrees Celsius. And we have to explain this result. So why was this? Because at 90 degrees Celsius, the enzyme would have been denatured and active site would have been deshaped or deformed. So the substrate would no longer be able to fit in the active site. So the answer to this question, you have to write the answer in this way, that beaker A produced, first of all, we will tell the results that beaker A produced more juice than beaker B. And then we will explain the results that beaker A was at 45 degrees Celsius, at which the enzyme was working at its best activity. And at beaker B, which was kept at 90 degrees Celsius, this denatured the enzyme and the substrate could no longer fit into the active site. Now, part C says bacteria can be genetically modified to produce enzymes for use in biotechnology. State 2 reasons why bacteria are suitable for this purpose. Bacteria are used to produce enzymes and they're used in biotechnology. Why is that so? Because seriously, there are no ethical concerns when we use bacteria and when we use viruses. All right. If we would have been using human genes, so therefore there would have been a lot of ethical concerns. All right. Moreover, there is a rapid reproduction, multiplication, or replication rate at bacteria because they carry out binary fission, they are genetically identical, and they have asexual reproduction. So this results in the rapid. If we add the bacteria in a fermenter and provide optimal temperature, they will easily create a lot of colonies and they will easily replicate themselves a lot of times. All right. So we can write down any 2 reasons from so many reasons which I have just explained it to you, that no ethical reason, no ethical concerns. Number second reason could be that they have rapid replication or reproduction rate. Some more points I'm writing right over here is that they produce identical cells. They are easy to grow. All right. Now, question number 6. Question 6 says figure 6.1 is a cross section of a leaf plant, like plant leaf. All right. So this is the leaf. This is a cross sectional diagram. So we have J, we have K, we have L. So they say identify each J, K, and L and explain the importance of each in the process of photosynthesis. So J, the topmost layer is the cuticle. Always remember that we have the cuticle on the top of the leaf. This is the cuticle. And then mesophil cells, which are known as mesophil cells. And then we have the spongy mesophil cells, which have the air spaces in between. Okay. And then we have the lower epidermis. All right. So we have to tell the role. of each of the structure over here. So the role of the cuticle is that they are transparent and they allow the light to pass through them, which enables the process of photosynthesis. All right. Then the K. The K is, as we talked about, that this is a palisade mesophyll cell. How did I came to know about this? Noted. I noticed the wait. I noticed the parallel shape of these cells. Alright, parallel shape of these cells showed that they are the palisade mesophyll cells. The palisade mesophyll cells have abundance of chloroplast and they have chlorophyll to absorb light. Alright, then L was lower epidermis. The lower epidermis always have the guard cells and they have stomata, which enables the gaseous exchange, the exchange between carbon dioxide and the oxygen. Alright, you can read the answer to this question. It's like I have just identified it in a word, the structure in a word, and then I have told the importance of each of their structures. All right. Now, part B says name and describe the function of one tissue found in vascular bundle of the leaf. So, vascular bundle comprises of two tissues, xylem or phloem. You can either talk about xylem or talk about phloem, and you have to name and then you have to describe the function. It is a 4-mark question. So, if you're talking about xylem, then obviously we all know that xylem is a hollow tube that is lignified and it enables the main function of xylem is to provide transpiration pull. Secondly, it enables the transport of water and mineral ions from the root in upward direction towards the leaf. And thirdly, we all know that the flow of water in the xylem is in one direction. And now let's talk about another vascular bundle. If you write down about phloem, then you have to write the name and describe its function as well. So the function of the phloem is translocation, and it transports sucrose and amino acids. Basically, it assimilates from source to sink or sink to source. So this means that the flow of the substances in the phloem is bidirectional, right? So you can either choose either one of them, any one of them, and write down the answer. Over here, I'm writing about xylem. So in the same way, you can also write about phloem that they carry out translocation, they carry out transport of assimilates from the source to sink and sink to source, and the flow of the direction of the flow is bidirectional. All right. So now part 7, question 7. Let's read question number 7. Mangrove trees grow in forests that form an important coastal ecosystem. Mangrove forests have a high biodiversity with many different animal species found in them. Describe what is meant by each of the following terms. So they are just asking the definitions of these words. So what is ecosystem? Ecosystem is basically the interaction of different organisms with each other, with the interaction with each other and with the non-living, the abiotic component of the environment. If we talk about biodiversity, the definition of biodiversity is that the number of different species in a particular area is known as biodiversity. If we talk about species, part 3 says species. So the species are the group of organisms that can reproduce to produce a fertile offspring. So these are the definitions, and you have to make sure that you use the exact terminology and you use the exact words that are mentioned in the mark scheme or that are mentioned in your syllabus. So you need to memorize these terminologies in order to score full marks. This is the answer to this question, and you can read it from here. So I hope the definitions are clear to you guys. And now moving towards part 3, like many forests around the world, mangrove forests are vulnerable to the effects of human activity. Explain how forests can be conserved as a sustainable resource. So this question is for 5 marks, and they're asking you how to conserve forests. So the question is about the conservation of forests. So there's a lot of points to answer in this question, and you can add like for example, first of all, anything to conserve anything, we need to provide awareness of its importance to the population, right? So first point you can write over here is that to spread education and spread or raise awareness regarding the importance of forest. And the second thing is that you have to stop the deforestation, or you have to make sure that there is a certain of particular amount or a particular cut off beyond which deforestation is prohibited. So to write down this thing that you can write down like this as generate or reinforce laws to make sure or to stop the deforestation and or cutting down of the trees. Moreover, always make sure to cut the trees from a selected areas. Obviously, we should not cut the trees from the areas that are populated or that are residential areas, because obviously that is going to create or give rise to the harsh or harmful environmental condition for the people living there, right? So secondly, always make sure that there is the replanting or reforestation activities as well before the deforestation is taking place. Alright, so like before cutting the trees, make sure to plant the trees to replace the loss that has to occur after cutting the trees. Alright, so these are the points you can include in your answer that raise awareness regarding the importance of forests and introduce quotas and laws to limit deforestation. And trees should be cut from specific areas, and we should start replanting trees before cutting of the trees to prevent the risk of deforestation and to ensure that trees do not was clear enough. And you don't have any questions. If you have any questions, you can always ask in the comment section below, and make sure to like, subscribe, and share the channel and the videos for the good cause of the people of the Palestine. Alright, so we'll meet in the next video with a new paper. Till then, goodbye and good luck.